Good morning to all the viewers. Today I have brought some science materials for SE students. Today's lesson is heredity. It is the chapter 4 of science of grade 10. This lesson is equally important for SE appearing students and the students who are appearing for supplementary examination. So heredity, today we are going to discuss about heredity. So this unit carries the marks, maximum 10 marks, 8 plus minus 10. So learning outcomes. After the completion of this unit, student will be able to describe the concept and importance of mitosis and meiosis cell division, introduce chromosome and gene, differentiate between DNA and RNA on the basis of their structure and function, and state the importance of DNA in different research works. Describe the role of sex chromosome in sex determination of human beings. Explain the Mendel's laws related to monohybrid cross. Introduce genetic technology and investigate about its implementation. So we have here some important lesson notes. Heredity. The inheritance of character from parents to offspring is called heredity. Cell division. The process of division of a mature mother cell into two or more daughter cells is called cell division. Types of cell division: mitosis cell division and meiosis cell division. There are basically two types of cell division. Mitosis cell division, it is the somatic cell division in which a diploid mother cell divides to form two diploid daughter cells. Importance of mitosis cell division: it helps in growth and development of the body, it helps in asexual reproduction, it helps in wound healing process, it maintains genetic stability. Meiosis cell division, it is the reproductive cell division in which a diploid mother cell divides to form four haploid daughter cells. Importance of meiosis cell division: it helps to form gametes, it helps in sexual reproduction, it causes genetic variation which helps in organic evolution. RNA, ribonucleic acid. DNA, deoxyribonucleic acid. Chromosome. Chromosome is the thread-like structure in the nucleus of cell that carries information from parents to offspring. Based on location and function, there are two types of chromosome. That is somatic chromosome. The chromosome that determines physical characteristics of an organism is called somatic chromosome. There are 22 pairs, that is 44 somatic chromosome in the human cell. Sex chromosome. The chromosome that determines the sex of an individual, gender of an individual, is called sex chromosome. There are one pair of sex chromosome, XX or XY, in human cell. Gene. The unit of heredity that transmits particular character from parents to offspring is called gene. Sex determination. The process of determining the gender of a baby on the basis of types of sex chromosome is called sex determination. Genetics. Genetics is the branch of biology that deals with the study of heredity and variation. Dominant characters. The character which dominates the other character and is expressed in first generation is called dominant character. Example: tall character in the cross of tall and dwarf. Tall is dominant character. Recessive character. The character which is suppressed in first generation is called recessive character. Example: dwarf character in the cross of tall and dwarf. Dwarf is recessive character. Phenotype. The external feature or external appearance of an organism is known as phenotype. Genotype. The genetically determined character is called genotype. Monohybrid cross. The cross between two organisms considering single unit of character is called monohybrid cross. Example: cross between tall and dwarf plant. Pure tall and pure dwarf plant. Hybrid. The offspring produced by crossing two genetically different parents is known as hybrid. Mendel gave different laws. Among them, first is law of dominance. Law of dominance states that when a cross is made between a pair of contrasting characters, only one character is expressed in first generation and that character is called dominant character. Law of segregation or purity of gametes. Law of segregation states that though the hybrid of first generation which contains two contrasting characters, they do not mix each other but separate out at the time of gamete formation. Genetic technology. Genetic technology is a modern branch of biotechnology that allows direct modification or removal of gene or transfer of gene from one species to another. DNA test. DNA test is a test that determines DNA of a person to study criminal activities, to identify the individual, or to show the peoples are relatives. Selective breeding. Selective breeding. The process of selecting the animals or plants with the best qualities and interbreeding them to produce offspring with the desired characteristics is called selective breeding. There are various methods of selective breeding. One is inbreeding, line breeding, self-pollination, crossbreeding, etc. Inbreeding. Inbreeding is the production of offspring by mating the organisms that are closely related genetically. Example: Siamese cat and Labrador retriever dog. Line breeding. It is a type of selective breeding in which cross is made between two distant relatives to produce the offspring with desired characteristics. Self-pollination is the process of transfer of pollen grains from anther to stigma of a flower within the same species of flower. Cross-pollination. The cross between two genetically crossbreeding. The cross between two genetically different and unrelated parents is called crossbreeding. Illustration of crossbreeding. Liger. Liger is a hybrid obtained by crossing male lion and female tiger. It behaves more like lion. Tigon. It is hybrid obtained by crossing male tiger and female lion. It looks like tiger but behaves like lion. Buffalo. Buffalo. It is the hybrid produced by crossing a bull male and buffalo female. Zebroid. It is the hybrid animal obtained by crossing zebra and horse. It lacks reproductive power. Mule, Khachar, and Nepali. It is the hybrid obtained by crossing a horse and donkey. It is sterile. Pomato. It is a hybrid obtained by crossing potato and tomato. Advantages of crossbreeding. It has to combine the desired features of two genetically different organisms. Human beings can produce organisms with desired features. Animals having better traits than parents can be developed. The immunity, strength, age, vigor, etc. of an organism can be improved by this method. Crop production can be produced from plants produced by this method. Crop production can be, we can say, increased from the plants produced by method. Disadvantages of crossbreeding. It may lead to problem in breeding policies in future due to its misunderstanding and mismanagement. The hybrid organisms are relatively very cheaper than purebred organisms that may lower the income of the farmers. It may lead to extinction of purebred animals or organisms. The natural traits of organisms may be declining due to this method. Artificial insemination, AI. Artificial insemination. Artificial insemination is the technique in which semen with the living sperm is collected from the advanced bred male and introduced into female reproductive tract at proper time with the help of instrument. It is also called intrauterine insemination, IUI technology. Advantages of artificial insemination, AI. There is no need of rearing male for breeding that saves expense of rearing. It helps to control the infection and spreading of diseases during mating. Collected semen can be used even after the death of the male. Collected semen can be transported over long distance for fertilization. This method increases the rate of fertilization. Disadvantage: artificial insemination. It requires all well-trained manpower and special equipment. It requires more time for its preparation than natural breeding. There can be infection due to lack of sterilization of equipment. Equipment. Equipment means instrument. Equipment means instruments. In vitro fertilization. IVF. In vitro fertilization is a medical procedure whereby an egg is fertilized by sperm in a test tube or elsewhere outside the body. An embryo thus formed is transferred to the mother's uterus after a few days. First test tube baby produced in Nepal is Om Mori Thamang. It's produced on 3rd March 2005. First test tube baby produced in the world is Louis Joy Brown. 25th July 1978. Advantages of IVF. It is boon to the couple who are unable to conceive the baby naturally. It allows conception by using couple's own sperm and ovum or by the use of donor's sperm and ovum. It helps to eradicate various genetic and chromosomal disorders. It increases the possibility of birth of a healthy child. Disadvantages of IVF. In vitro fertilization. Disadvantages. This method has lower success rate of bearing a child. There can be problem of multiple fertilization as well. Multiple fertilization. It is costly technology and cannot be afforded by common people. The problem of early birth and underweight birth may arise. Multiple choice question from this lesson. Heredity. Section A. Carrying 1 marks. Choose the best alternative for the following questions. What is the unit of heredity? Unit of heredity is obviously gene. Gene is the correct answer. What is the chromosome made up of? Chromosome is obviously made up of DNA and protein. How many chromosomes are there in a human cell? Obviously there are 46 chromosomes. How many sex chromosomes are there in human cells? One pair, that's 2. What is the function of somatic chromosome? Somatic means body chromosome, so it helps in physical growth of body. What is the function of sex chromosome? Sex chromosome helps to determine the sex or gender of a baby. Which of the following is the correct for mitosis cell division? Mitosis cell division, it helps in wound healing. It helps in wound healing process. Which of the following is affected by high intensity radiation? Obviously DNA is affected by high intensity radiation. Which of the following process is affected by lack of DNA transcription? Lack of DNA transcription affects the protein synthesis. Protein synthesis. Which of the following represent nitrogen base in DNA? DNA contains adenine, thymine, guanine, cytosine. So ATGC is the correct answer. A nucleoside is composed of a base and a sugar. Nucleoside consists of a base and sugar. Which of the following is found only in RNA? Uracil. That nitrogen-based uracil is found only in RNA. In DNA, thymine is found, only thymine, but in RNA, the uracil is found. What effect is seen in the baby of living being by the destruction of nucleotide during DNA replication? Its correct answer, it causes genetic disorder. Which type of cell division help in asexual reproduction? Asexual. Mitosis helps in asexual reproduction. Which of the following figure represents mitosis cell division? Mitosis cell division gives diploid cell dividing to form two diploid cells, so from 2n to 2n. So this is the correct answer. The mitosis cell division is a reductional cell division. Sorry, the meiosis cell division is called reductional cell division because reduction means chromosome number is reduced. Number of chromosomes decreases in the shell during meiosis. Yes, this is the correct answer. A statement and two arguments are given below. Statement: mitosis cell division is called equational cell division. It's correct. It involves division of diploid cell to form two diploid cells. This is also correct. It occurs in germ cell. Germ cell means reproductive cell. That is gametes. This is not correct. Now here is option. Statement and argument 1 are correct, but argument 2 is correct. Statement and argument incorrect, but argument 2 is correct. This is not right. Statement and argument 1 are correct, but argument 2 is incorrect. This is right. Because these two statement and arguments 1 are correct, but argument 2 is incorrect. So option B is correct answer. Which of the following is first stage of mitosis? Obviously prophase. Gamete formation takes place through which process? Obviously meiosis process. Karyokinesis means it is the division of nucleus. Which of the following indicates the set of chromosomes in the cell of a woman? Woman contains XX chromosome only, XX. So 44 plus XX, altogether 46 in human. Cytokinesis is the division of cytoplasm. Crossing over occurs during Peykatin stage of meiosis cell division. Which of the following represents baby boy? So XY, there should be XY, and in human being there are 46 chromosomes, so 44 plus XY, 46. Which of the following is correct statement? Sperm contains X chromosome and ovum contains only Y chromosome. This is not wrong. Sperm contains Y chromosome and ovum contains only X chromosome. This is also not right. Sperm contains X or Y chromosome, ovum contains only X chromosome. This is the correct answer. Sorry. Who is known as father of genetics? Obviously Gregor Mendel. The plant selected by Mendel for his experiment is pea plant, Pisum sativum. The genetic composition of a living beings is called genotype. The alternative form of gene is called allele. How many characters of pea plant were studied by the Mendel? 7 pair of contrasting characters were studied by Mendel. 7. What is the phenotype ratio of monohybrid cross? Obviously 3:1. What is the genotype ratio of monohybrid cross? Monohybrid. 1:2:1 is correct answer. Which of the following represents hybrid organism? Hybrid contain both characters, dominant and recessive. Here also dominant, recessive, capital small. So these two are correct. It means both A and B are correct. Which of the following is dominant character? The axial flower is dominant character. What is called the character that is transmitted from one generation to another generation? Obviously, it is the hereditary character. The character that is hidden in first generation of monohybrid cross is called recessive character. That is hidden is recessive character. White offspring is produced from black parents. What does it mean? It means parents are hybrid. So if the parents are hybrid, it may contain black as well as white characters. That's why. So though the black is dominant character and parents look like dominant externally, but they have white character as well as a recessive character. So white offspring are produced in second generation. So correct answer is parents are hybrid. The cross between advanced boar male goat with a local female goat is selective breeding. Advanced boar male goat with the female local goat is the advanced goat. This is the female local goat. Selective breeding. The new plant produced by crossing orange and lemon is hybrid plant. Which of the following is correct for reproductive power of a mule? Mule is sterile, means it is not fertile, it cannot reproduce. Now section B, very short question, 1 marks. What is heredity? The inheritance of character from parents to offspring is called heredity. Define cell division. The process of division of mature mother cell into 2 or more daughter cell is called cell division. What are different types of cell division? The types of cell division are mitosis cell division and meiosis cell division. What is meant by mitosis cell division? It is the somatic cell division in which a diploid mother cell divides to form 2 haploid daughter cells. Diploid mother cell division divides to form 2 diploid daughter cell. Mitosis, here is figure for this also. Write the type and site of cell division shown in the figure. It is mitosis cell division that occurs in somatic cell. This. Define meiosis cell division. It is a reproductive cell division in a diploid mother cell divides to form 4 haploid daughter cells. This is the correct answer. What type of cell division is shown in the figure? Right, it's one important. This figure is for down question. It is meiosis cell division. It helps in sexual reproduction. What are different phases of mitosis cell division? The different phases of mitosis cell division are prophase, metaphase, anaphase, and telophase. What is the full form of DNA and RNA? Sorry, RNA and DNA. RNA ribonucleic acid, DNA deoxyribonucleic acid. Define the following term: chromatid, spindle fiber, centromere, karyokinesis, cytokinesis. Chromatid, 2 symmetrical structure of chromosome is called chromatid. Spindle fiber is a thread-like structure which is attached to the centromere of chromosome in metaphase stage. Centromeres. The 2 chromatids of chromosome is joined to each other by a beaded structure which is called centromere. Karyokinesis is the process of division of nucleus during cell division. The division of nucleus is called karyokinesis, whereas division of cytoplasm is called cytokinesis. Define chromosome. Chromosome is thread-like structure in the nucleus of cell that carries information from parents to offspring. What is chromosome composed of? Chromosome is composed of DNA and protein, histone protein. Label parts A and B of the given figure. A is chromatid, so this 2, chromatid 1, chromatid 2, 2 chromatid, and this beaded-like structure where the chromatid are attached, this is called centromere. Centromere means this round structure. Give two examples of nucleic acid. Two examples of nucleic acid, DNA and RNA. What is somatic chromosome? The chromosome that determines physical characteristics of an organism is called somatic chromosome. How many somatic chromosomes are there in human cell? There are 22 pairs somatic chromosomes. 44, that is somatic chromosome in human cell. What is the number of chromosomes in the cell of woman? The number of chromosomes in woman cell is 44 plus XX. Define sex chromosome. The chromosome that determines sex of an individual is called sex chromosome. There is one pair of sex chromosome, XX or XY in human cell. XY in male, XX in female. Define the following term. Synapsis. Synapsis is the process of pairing of homologous chromosomes. Crossing over. The process of exchange of genetic material between the chromatids of homologous chromosome during Pectine stage. Homologous chromosome during Pectine stage of meiosis. The process of exchange of genetic material between the chromatid of homologous chromosomes during Pectine stage of meiosis. One is called crossing over. The homologous chromosome, the set of identical chromosomes that have come from male and female parents, are called homologous chromosome. Diploid cell, the cell which contains full number, two set of chromosome is called diploid cell. It is zygote. Haploid cell, the cell which contains half number n of chromosome is called haploid cell, e.g. sperm, egg, etc. Nucleoside, the single unit of DNA consisting of nitrogen base and deoxyribose sugar is called nucleoside. Nucleotide, the single unit of chromosome consisting of a nitrogen base, deoxyribose sugar and phosphate is called nucleotide. What is DNA made up of? DNA is made up of nitrogen bases, deoxyribose sugar and phosphate molecule. Write the function of DNA. The function of DNA are: it transmits hereditary character from parents to offspring, it transcripts RNA. Write the function of RNA. The function of RNA are: it helps in protein synthesis, it controls metabolic activities. What are different types of RNA? The different types of RNA are messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA. What is composition of RNA? RNA is composed of nitrogen bases, ribose sugar, and phosphate molecule. What are nitrogenous bases present in DNA? What are nitrogen bases present in DNA? They are adenine, thymine, guanine, and cytosine. What are nitrogen present in RNA? Nitrogen present in RNA are adenine, uracil, guanine, and cytosine. Uracil, guanine, and cytosine. Mention the difference between RNA and DNA and RNA in terms of nitrogen bases present in them. DNA contains thymine, whereas RNA contains uracil as a nitrogen base. ATGC. What do ATGC and AUGC stand for? ATGC stands for adenine, thymine, guanine, cytosine. AUGC stands for adenine, uracil, guanine, and cytosine. Mention the difference between DNA and RNA in terms of sugar present in them. DNA contains deoxyribose sugar, whereas RNA contains ribose sugar. That is the main difference. Where is DNA commonly found? DNA is commonly found in chromosome present in the nucleus. Where is DNA found in prokaryotic cell in virus? DNA is found in cytoplasm in prokaryotic cell and inside capsid in virus. Capsid in. What is centromere or kinetomer? The two chromatids of chromosome are joined together at a point called centromere. What is the significance of crossing over? It causes genetic variation in organisms that leads to organic evolution. Define gene. The unit of heredity that transmits particular character from parents to offspring is called gene. Define sex determination. The process of determining a gender of a baby on the basis of types of sex chromosome is called sex determination. Define genetics. Genetics is the branch of biology that deals with the study of heredity and variation. What is dominant character? The character which dominates the other character and is expressed in first generation is called dominant character. Example, tall character in the cross of tall and dwarf is the dominant character. What is recessive character? The character which is suppressed in first generation is called recessive character. Example, dwarf character in the cross of tall and dwarf. Define phenotype. The external feature of an organism is known as phenotype. What is genotype? The genetically determined character is called genotype. What is monohybrid cross? The cross between two organisms considering single unit of character is called monohybrid cross. Example, the cross between tall and dwarf plants. Define hybrid. The offspring produced by crossing two genetically different parents is called hybrid. What are Mendel's three laws in genetics? Mendel's three laws in genetics are law of dominance, law of segregation, and law of independent assortment. State law of dominance. Law of dominance states that when a cross is made between a pair of contrasting character, only one character expressed in first generation, that character is called dominant character. State law of segregation. Or purity of gamete. Law of segregation states that though the hybrid of first generation which contains two contrasting characters, they do not mix each other but separate out at the time of gamete formation. Write any two dominant character of human beings. Two dominant character of human beings are free earlobes and dimple cheeks. That are the two dominant characters. Write any two recessive characters seen in human beings. None curly hair and none dimple cheeks. What is genetic technology? Genetic technology is a modern branch of biotechnology that allows direct modification, removal of gene, or transfer of gene from one species to another. Define DNA test. DNA test is a test that determines the DNA of a person to study criminal activities, to identify the individual, or to show the people are relatives. Okay. What is the use and purpose of DNA testing? Actually, DNA testing is used for identify the species of fossils, identify male or females, diagnosing genetic disorder, criminal investigation, and making vaccine, etc. What is selective breeding? The process of selecting the animals or plants with the best qualities and interbreeding them to produce offspring with the desired characteristics is called selective breeding. What are different methods of selective breeding? Inbreeding, line breeding, self-pollination, crossbreeding. Define inbreeding. Inbreeding Inbreeding is the production of offspring by mating the organisms that are closely related genetically. For example, Siamese cat and Labrador Retriever dog. These are examples of offspring produced by inbreeding. Siamese cat and Labrador Retriever dog. Define line breeding. It is a type of selective breeding in which cross is made between two distant relatives to produce offspring with desired characteristics. Define self-pollination. It is the process of transfer of pollen grain from anther to stigma of a flower. What is crossbreeding? Crossbreeding. The cross between two genetically different and unrelated parents is called crossbreeding. What is liger? Liger is the hybrid obtained by crossing male lion and female tiger. It behaves more like lion. What is tigon? Tigon is a hybrid obtained by crossing male tiger and female lion. It looks like tiger but behaves like lion. What is buffalo? The hybrid obtained by crossing bull means male and buffalo female is known as buffalo. What is zebroid? It is the animal obtained by crossing zebra and horse. It lacks reproductive power. What is mule culture? It is a hybrid obtained by crossing horse and donkey. It is sterile. What is pomato? It is a hybrid plant obtained by crossing potato and tomato. What is artificial insemination? Insemination. Artificial insemination is a technique in which semen with the living sperm is collected from the advanced bred male and introduced into female reproductive tract at the proper time with the help of instrument. It is called intrauterine insemination (IUI). What is in vitro fertilization (IVF)? In vitro fertilization is a medical procedure whereby an egg is fertilized by sperm in a test tube or elsewhere outside the body, and embryo thus formed is transferred to the mother's uterus after few days. With the first test tube baby in Nepal, Om Modi Thamang. With the first test tube baby in the world, Louis Joy Brown. What are sub-stages of prophase 1 of meiosis cell division? Just for extra knowledge. Prophase 1 divided in following 5 sub-stages: leptotin, zygotin, pectin, diplotin, and diakinesis. Short question, 2 marks, section B. Mention importance of mitosis cell division. It helps in growth and development of body. It helps in asexual reproduction. It helps in wound healing process. It maintains genetic stability. Enlist the importance of meiosis cell division. It helps to form gametes. It helps in sexual reproduction. It causes genetic variation which helps in organic evolution. Enlist the advantages of crossbreeding. It helps to combine the desired feature of two genetically different parents. Human beings can produce organisms with desired features. Animals having better traits than parents can be developed. The immunity, strength, age, vigor, etc. of an organism can be improved by this method. The crop production can be increased from the plants produced by this method. Enlist the disadvantages of crossbreeding. Crossbreeding has following disadvantages. It may lead to problem in breeding policies in future due to its misunderstanding and mismanagement. The hybrid of organisms are relatively very cheaper than purebred organisms that may lower the income of farmers. It may lead to extinction of purebred organisms. The natural traits of organisms may be declining due to this method. What are the importances of artificial insemination? What are the points of importance of artificial insemination? There is no need of rearing male for breeding, then that saves the expenses of rearing. It helps to control infection and spreading the disease during mating. Collective semen can be used even after the death of the male. Collective semen can be transferred over a long distance for fertilization. This method increases the rate of fertilization. What are the disadvantages of artificial insemination? Artificial insemination. Disadvantages of artificial insemination. It requires well-trained manpower and special equipment. It requires more time for its preparation than natural breeding. There can be infection due to lack of sterilization of equipment. Lack of equipment. Here is some error. Equipment. Differentiate between autosome and sex chromosome. Autosome and sex chromosomes. Autosomes. These are the chromosomes that determine the physical traits of the body. These are the chromosomes that determines the gender of the person. There are 22 pairs of autosomes in a human cell. There is one pair of sex chromosome in a human cell. Mitosis and meiosis. It is division of diploid cell to form two diploid cells. It is division of a diploid cell to form four haploid cells. It helps in physical growth and development body. It helps in the formation of gametes and sexual reproduction. It occurs in somatic cell. It occurs in reproductive cell. It maintains genetic stability. It causes genetic variation. Difference between DNA and RNA. It has double-stranded structure. It has single-stranded structure. It helps in protein synthesis. It helps in transmission of character from parents to offspring. It has been opposite. This point should be here. Yes, actually DNA helps in the transmission of character from parents to offspring, and RNA helps in protein synthesis. It contains thymine as nitrogen base. It contains uracil as a nitrogen base. That's correct answer. So DNA double-stranded structure, RNA single-stranded structure. DNA helps in transmission of character from parents to offspring. RNA helps in protein synthesis. It contains thymine as nitrogen base. It contains uracil as nitrogen base. Haploid cell and diploid cell. Haploid cell. It contains half or n number of chromosomes. It contains full or 2n set of chromosomes. The haploid cells are produced by meiosis, example sperm egg. Diploid cells are produced by mitosis, example zygote. Dominant and recessive character. Dominant, it is the character which is expressed in first generation. It is the character which is suppressed in first generation. Tall is dominant character in the cross of tall and dwarf. Dwarf is recessive character, not dominant. It is recessive, R-E-C-E-S-S-I-V-E. It is recessive character in the cross of tall and dwarf. Phenotype and genotype. It is the external feature of an organism. It is genetically determined feature of an organism. Organism with the same phenotype can have different genotypes. Organisms with same genotype have the same phenotype. So organisms with same phenotype may have different genotypes. The organisms with same genotype have the same phenotype. Inbreeding and crossbreeding. The act of breeding between male and female who are closely related genetically is called inbreeding. If a purebred male and purebred female of two different species of the same genus are crossed to produce new offspring, it is called crossbreeding. Crossbreeding is the cross between two genetically different parents is crossbreeding. It decreases the gene pool of an organism. It increases the gene pool of organism. Artificial insemination and in vitro fertilization. Artificial insemination and in vitro fertilization. Artificial insemination. It is the deliberate introduction of sperms into a female cervix or uterus with the help of instrument. It is the process in which ovum and sperm are fertilized outside the female body in the test tube and then embryo is implanted in the uterus. Sperm is put inside uterus and embryo is put inside uterus in vitro fertilization. Tigon and liger. The hybrid obtained from a male tiger and female lion is called tigon. The hybrid obtained from male lion and female tiger is called liger. It is smaller than parents. It is bigger than parents. Anaphase of mitosis and anaphase 1 of meiosis. It's for extra knowledge. Centromere divides into two halves. Centromere does not divide into two halves. Number of chromosomes remains same in it. Number of chromosomes is reduced to half in it. Mitosis and meiosis cell division. It is the division of diploid cell to form two haploid cells. It is the division of diploid cell to form four haploid cells. It occurs in somatic cell. It occurs in reproductive cells. Crossing over does not occur in mitosis. Crossing over occurs in meiosis. It maintains genetic stability. It causes genetic variation. Differentiate between DNA and RNA in terms of sugar and nitrogen base present in them. DNA, it contains deoxyribose sugar. RNA, it contains ribose sugar. DNA, it contains thymine as a nitrogen base. It contains uracil as a nitrogenous base. What is nucleic acid? Write its type. Nucleic acid are naturally occurring chemical compound in living cells that carry genetic information. There are two types of nucleic acid, namely DNA and RNA. Clarify the role of chromosomes in the body of living beings. Chromosomes have the following roles. It transmits parental character to offspring. It carries codes for physical structure of body of living organisms. It determines the gender of a person as well. It also transmits genetic diseases and disorders. Give reason for the following questions. Offspring have same character as their parents. Offspring have same character as their parents because they inherited hereditary character from their parents, for which genes play the vital role in the transmission of parental character to offspring. Offsprings experiences. In this way, inheritance of parental character to the offspring by the gene is the main reason behind the resemblance of offspring to their parents. The male has a main role in the determination of sex. Of course, females have only X chromosome in their egg cells that they produce typically. Female have only X chromosome in their egg cells. This is not so important. While the male sperms can carry either X or Y. Male sperm cell can carry either X or Y. During fertilization, female always gives X chromosome, and if male gives X chromosome, a baby girl is born. Baby girl is born. And if male gives Y chromosome if male gives Y chromosome, a baby boy is born. Therefore, male has main role in the determination of sex. Female has always X chromosome, that is XX chromosome. Male have XY chromosome. So if male gives X chromosome during fertilization, daughter is born. If male gives Y chromosome during fertilization, boys, that is son is born. Next question. Though males have both X and Y chromosomes, sex chromosomes, some of them have only male or only female kids. Though male have both X and Y sex chromosomes, some of them have only male or female kids because male sperm containing only one type of chromosome, either X or Y, might have fertilized the female egg containing always X chromosome by coincidence. This is just a coincidence. If during fertilization only X chromosome from male sperm goes, then the female baby is born, and if Y chromosome only goes during fertilization by coincidence, then male baby is born. It is just coincidence. Mitotic cell division is also called reductional cell division because number of chromosomes is reduced to half in daughter cell compared to mother cells. Mitotic cell division is called equational cell division. Equational cell division because number of chromosome in daughter cell is equal to number of chromosome in the mother cells. Sexual reproduction is impossible without meiosis or meiotic cell division. Sexual reproduction is impossible without meiosis cell division because male gamete and female gamete are needed for reproduction, and these gametes are produced from meiosis cell division. Meiosis cell division brings variation. Variation is difference seen between two individuals in terms of appearance and genetic structure. Meiosis cell division brings variation because genetic material that have come from father and mother are exchanged during crossing over in the first karyokinesis of meiosis cell division. Children look like their parents but not exactly the same. Children look like parents because there is inheritance of parental character to the offspring by the genes, but children are not exactly the same as their parents because genetic material that have come from father and mother are exchanged during crossing over of meiosis cell division. Mendel selected pea plant for his experiment because pea plant has a close bisexual flowers. It helps in self-pollination. It has a very short life cycle, so that result can be obtained frequently. It has lots of its variety, observable contrasting character as well. Next question. When a tall pea plants and dwarf pea plants are cross-pollinated, tall plants are produced in first generation because tall is dominant character which dominates the recessive character. That's why. When cell breeding is done between hybrid, different types of offspring are produced. Because hybrid contains both dominant and recessive genes, these genes do not mix other according to law of segregation. So when self-breeding is done between hybrid, different types of offspring containing dominant character, recessive character, and hybrid character are produced. DNA testing is a reliable technique for criminal investigation. Yes, it is true. DNA testing is used to identify a person accurately. So if there is any material collected from the crime scene and that contain DNA, this testing can help to identify the criminal exactly. The samples from the crime scene such as semen, blood, sweat, nail, hair, saliva, etc. that are sent to laboratory and criminals are identified exactly based on the result. DNA testing of the suspect is also done. Two reports are compared to see if the suspect is guilty or not. Therefore, DNA testing is a reliable technique for criminal investigation. Genetic engineering involves the detailed study of DNA. The gene present in DNA contain hereditary information. Through the information of the structure of DNA, the function of DNA, the chemical composition of DNA, construction of DNA, molecular bonds in DNA, and any other information about DNA. Genetic engineers can cut, merge, transfer, combine, insert, or even artificially create DNA. This technology can be used in medicine, agriculture, animal husbandry, gene therapy, etc. In this way, genetic engineering involves detailed study of DNA. Offspring produced by crossbreeding may be sterile. Offspring produced by crossbreeding may be sterile due to difference in genetic makeup of parent species. Different species often have different number of chromosomes. When these species mate, their chromosomes may not pair up correctly during meiosis, leading to incomplete set of chromosomes in the offspring and hence sterility. Mule is sterile. A mule hybrid between a horse which has 64 chromosomes and a donkey 62 chromosomes contains 63 chromosomes. The odd number of chromosomes in mules prevents the proper pairing of chromosomes during meiosis, leading to sterility. Special attention should be given while collecting sample for DNA testing. Yes. Special attention should be given while collecting sample for DNA testing because any impurities or contamination in the sample could lead to wrong or unfair results. But if the special attention is given to sample, it helps to increase the accuracy of DNA testing. Gamete is haploid but zygote is diploid because gamete contains half number of chromosomes, only n number of chromosomes, but zygote contains double or full set of chromosomes, 2n. That's why zygote is diploid. Offspring produced by sexual and asexual reproduction are dissimilar because offspring produced by sexual reproduction contains the gene of both the parents, whereas offspring produced by asexual reproduction contains gene of only one parents. Meiosis helps to maintain fixed number of chromosomes in the organism. Meiosis is a division of haploid cell to form four diploid cell to form four haploid cells. With this haploid gametes are produced which on fertilization forms a diploid zygote. This prevents doubling of chromosome number from generation to generation. So meiosis helps to fix the number of chromosomes in an organism. Blue-eyed offspring may be produced from brown-eyed parents. As brown eye is dominant character, blue eye is recessive character. Blue-eyed offspring may be formed from the brown-eyed parent when both the parents are hybrid brown. According to law, purity of gamete, two contrasting characters contained in the hybrid do not mix each other. They separate out during gamete formation and express in next generation. So blue-eyed offsprings may be formed from the brown-eyed parents. Both the parents are hybrid with brown-eyed, having blue-eyed as a recessive character. Both the parents are hybrid with brown-eyed, having blue eyes. This point is not so important. What will happen if meiosis cell division does not occur in the reproductive cell of an organism? If the meiosis cell division does not occur in reproductive cell of an organism, then the organism will be either sterile or number of chromosomes would be double each time they reproduce. Meiosis cell division is essential for the formation of male and female gametes. If the meiosis cell division does not occur in germ cell, either male or female gamete will not be formed. Offspring could not be produced, or if produced, the number of chromosomes would be doubled in the offspring. Clearly explain the role of gene in the transmission of hereditary character in the organism. Obviously, gene carries the vital role in the transmission of the characters. All the hereditary characters of an organism are stored by the gene. Gene controls the morphology of an organism. The gene controls growth and development of body parts of the organism, while some genes control the shape of face, color of skin, eyes, hair, etc. The characterized genes are transmitted from mother cell to daughter cell during formation of gametes, and the fusion of male and female gametes collect all the information in zygotes. Some variation caused by crossing over are also transmitted over generation. How is sex determined in human being? Explained in the chart. So the father has XY chromosome, mother has XX chromosome. If X combine with X, it forms daughter. If Y combine with X, it forms son. So there is 50% chance of son and 50% chance of daughter. So here is something explained. You can read yourself as well. If a woman is pregnant, what is the probability of giving birth to a daughter? Right, in percentage. So there is 50% chance of giving birth. 50% male, 50% female chance. So here is same thing written. You can read yourself. Question number 16. A couple gave birth to an only son. Does it mean that the testes of those male produce sperm having Y chromosome only? No, the produced sperm may not have Y chromosome only, rather it may have S chromosome too. The sex of newly born baby depends upon the possible combination of sex chromosome present in the gametes of parents, but by coincidence, each time Y chromosome of male gamete fuses with S chromosome of female gamete during fertilization, and male baby might have been born. Complete the following concept map. Reproductive cell, what? Meiosis cell division occur and form gamete. Somatic cell, what? Mitosis cell division and somatic daughter cells are formed. Difference between above process, mitosis and meiosis, already explained above. Give some example of genetic technology. There are various examples of genetic technology. Some examples of genetic technology are production of medically important products, including human insulin, human growth hormone, hepatitis B vaccine, as well as development of genetically modified organism, mutant organism, etc. Importance of genetic gene in genetic technology. DNA has great importance in genetic technology. So you can learn the importance of DNA in genetic technology from here. Explain the importance of genetic engineering. Following points enlightens the importance of genetic engineering. Various genetic technologies like transgenic technology, gene expression, and molecular markers technology etc. are used in agriculture. DNA testing is used in solving criminal cases and identifying parents and ancestors. Genetically modified crops and animals are used in agriculture and animal husbandry. Gene therapy is used to treat various genetic disorders. Explain Mendel's law of dominance and law of purity of gametes. We have already stated these two laws previously also. So contrasting and law of dominance, we have already learned this, and law of segregation also we have already learned this. Contrasting, so you can learn again. Now question number 20, are Mendel law also applicable to human beings? State any two difficulty Mendel law that we have faced if it is applied to human beings instead of pea plant. Of course Mendel law are also applicable to human beings, but you would have faced various difficulties if the experiment is carried out in human beings. Like cross-fertilization between selected male and female would not be possible due to social norms. Human beings have no lots of varieties like pea plants. Lifespan of human being is long, unlike pea plants, so result could not be obtained frequently. Question number 20. After mating of a black dog with a brown, all offspring in first generation were black. Why was brown colored dog not born? Because black is dominant character, which dominate the brown character, so only black is seen according to law of dominance. What is monohybrid cross? So in filial chart, the result obtained by crossing first and then self-pollinating the red flowering plant and white flowering plant, first and second generation. The cross between two organisms considering single pair of contrasting character is called monohybrid cross. We have already done this. Now here is pure red flower, pure white flower, cross fertilization. It gives all 100% hybrid here, because it's just like algebraic multiply, r multiply small r multiply small r, this way. Now self fertilization between two hybrid obtained from first generation. So there is possibility of pure hybrid capital RR, two are capital small and one is small small, because capital multiply capital gives capital capital, capital multiply small gives capital small, small multiply capital gives capital small, small multiply small r gives small small. So small small is pure white, capital capital is pure red, and mixing is hybrid. Phenotype ratio 3:1, genotype ratio 1:2:1. So in first filial chart, the result obtained by cross-fertilizing first and then self-fertilizing of long-winged Drosophila and short-winged Drosophila. Same like previous questions. Also write the genotype and percentage of long and short. This is the first generation where 100% hybrid long are obtained, and this is second generation where mixed type obtained, pure long, pure short, and hybrid long. Phenotype ratio 3:1, genotype ratio 1:2:1, percentage of long wing 75, percentage of short wing 25, because out of 4, 3 is long means 75, out of 4, 1 is short means 25%. The above chart can be expressed in table as well. So it is pure tall plant and pure dwarf plant where first cross fertilize and then self fertilize. The table below shows the result of this experiment. This is hybrid TT, capital small, so it's hybrid male, hybrid female. We can consider which filial generation does result belong to. Obviously, it is second filial generation. What is the percentage of plants having phenotypically tall character? Phenotypical tall, these three, capital T, phenotypically tall. So 75%. Out of 4, 3 means 75%. What is the pair of gene? Which pair of genes indicate pure dwarf plant? Small tt indicate pure dwarf plant. Let's consider black B is a dominant phenotype and white B is recessive in a cat. In a certain experiment, a black cat of genotype BB is crossed with the white cat of genotype BB. Result show with the help of table that ratio of black babies and white babies will be 1:1 on crossing them. The following table is obtained by crossing between cats of genotype BB and BB. Capital small here, small here. This result is obtained. This into this here, this into this here, this into this here, this into this here. The BB represent black and BB represent white character. So the ratio of black and white babies is equal to 2:2 is equal to 1:1. Explain with an example of man-made experiment can be done not only in plants but also animals. Yes, it is true that Mendel experiment can be done not only in plants but also in animals, as both parents and animal are living beings. Animal also contain allele of genes like plants. For example, TT represents pure tall and TT represents pure dwarf allele of genes of plant, and in the same way BB represents black mouse and BB represents white mouse. Like T and T represent dominant recessive character in plants, in the same way B and B represent dominant and recessive character in the animal mouse. Hence Mendel law are followed both on plants and animals. This is a logic. The character which is not seen in F1 generation is found expressed in F2 generation after crossing the hybrid of F1 generation. How? This is according to law of segregations, because law of segregation says that hybrid contains two different characters. They do not mix each other during though they are kept for a long time, but they are separated out at the time of gamete formation. That's why. Same thing written here. You can also read. Round-seeded pea plant and wrinkle-seeded pea plant are cross-pollinated first and then self-pollinated, and again the result obtained of second filial generation in the table. Answer the following question. RR and RR, hybrid cross, result obtained this way. One is pure what is called round, another is pure wrinkle, two are hybrid round. What is the ratio of plants showing dominant and recessive character? Ratio is 3:1. Write the genotype and phenotype of this generation. Genotype and phenotype ratio. Genotype ratio is 1:2:1. Phenotype ratio is 3:1. How genotype is 1:2:1? Because 1 is pure and 2 is hybrid and 1 is again pure wrinkle. So this. Among them, which plant is pure round seed and why? Pure round seed. RR, capital R is pure round seed, because both chromosomes are same and dominant, and pure round is dominant character. Pure red flower plant and hybrid red flower plant are cross-pollinated, which gave the result shown in the table below. Now answer the following questions based on the table. RR. Write the genotype and phenotype ratio according to the table. Genotype ratio pure red and hybrid red. pure red flower and hybrid flower are crossed. These two are pure, these two are hybrid. This and this come, this and this. Genotype ratio of pure red and hybrid red is 2:2. 2:2, and upon cancelling it gives 1:1, because pure red, 2 hybrid red, phenotypically 100% red, because there is capital R in all, so phenotypically they appear as red, so 100% red. Why is white flower plant not seen in the result? White flower plant not seen in the result because R, red, is dominant character. Which dominates the recessive small r character. When a cross is made between a black Guinea pig and white Guinea pig, the offspring of first filial generation were all black. Explain why white Guinea pig did not appear in first generation. This is also according to law of dominance. When a cross is made between a black guinea pig and white guinea pig, the offspring of first filial generation all black. The white guinea pig did not appear because the white color is recessive character of guinea pigs, which is suppressed in first generation, and only dominant characters in first generation. A teenage girl who lost her mental balance became a victim of rape and gave birth to a child. How can father of her child detect it? DNA testing is used to determine the father of the child. DNA test is proper method. How does cytokinesis occur in animal cell and plant cell? For extra knowledge, cytokinesis occurs by cell plate method in plant cell, whereas cleavage or constriction method in animal cell. Constriction or form method also it is called. Now time for section D. It has been long time. Let's go a little bit faster. Section D, long question is carrying for explain the importance of mitosis, cell division in the growth and development of body. So mitosis, cell division has the following importances. So here is the importance. You can learn yourself. Everything is written clearly here. Explain the role of mitosis and meiosis in the reproduction of organisms. Actually, both cell division is required for the reproduction of organism. Both mitosis and meiosis are important for the reproduction of organism. Meiosis basically helps in sexual reproduction by the formation of male and female gametes. The meiosis cell division in the germ cell of male gonads forms the male gametes, and the female gonads form female gametes. Meiosis cell division produces a haploid set of chromosomes in daughter cells. Then, after fusion of male gamete and female gamete during fertilization, zygote. Then, after fusion of male and female gamete during a zygote can again have diploid chromosome. Mitosis cell division basically helps in asexual reproduction of unicellular organisms. For example, some bacteria and amoeba reproduce asexually with the help of mitosis cell division, or diploid mother cell will split into diploid daughter organisms. Furthermore, repeated mitosis cell division in a single cell zygote. Furthermore, repeated mitosis cell division occurs in the single celled zygote, and growth and development of body of organism takes place. In this way, there is a great role of both mitosis and meiosis in the reproduction of organism. The District Animal Development Center conducted a camp to fertilize many cows at once. Which technique did that organization adopt at this time? Explain the technique in brief. The District Animal Development Center must have used artificial insemination (AI) technology to fertilize many cows at a time. Artificial insemination is the technique in which semen with living sperm is collected from the advanced bred male and introduced into female reproductive at proper time with the help of instrument. The concept of artificial insemination is to introduce sperm into female cervix or uterus. So for artificial insemination, healthy male gametes from the desired breed of animals should be collected. The male gametes should be washed and stored in the sperm bank. Then, when required, vet doctor will bring it to the farmer. Then the veterinary doctor will keep it in a sterilized syringe and use the syringe to place the male gamete near the cervix uterus of female animal. Hence, artificial insemination is used for the fertilization of animals. Is genetic engineering a boon or ban or cause for present era? Give your arguments. Genetic engineering is a great achievement of present era which is justified by following points. Different genetically modified crops have produced enough food to feed billions of people throughout the world to fulfill the demand of increasing population. The genetic engineering is widely used to manufacture insulin, vaccine, and many medical technologies that are saving millions of lives every year. DNA testing is genetic technology. It has helped us to solve thousands of criminal cases. Gene therapy is used to treat genetic disorder, chromosomal disorder, etc. So genetic engineering is a boon, not a ban for present era. IVF is proved to be a boon for childless couples. Justify the statement. When there is some problem in the fallopian tube, underdevelopment of ovary, testes, genetic disorder, or radiation effects, the married couple can't give birth to baby. The people of the society talk more about those people lacking children. It could stress them. However, in vitro fertilization is a very scientific assisted reproductive health program. Through IVF fertilization, male sperm and female ovum can be done in a test tube and later transplanted to mother uterus. As a result, infertile couple can give the birth to the baby and share the happiness. For all these reasons, IVF technology can be regarded as boon to the childless couples. How has AI technology helped to bring happiness to the farmer? Explain. Obviously, AI technology brings happiness to the farmers because it has reduced the burden and saved the expenses of keeping male-bred animals. The farmer can freely choose the semen of advanced male-bred animal of their choice. It is cheaper means of reproduction for farmers. This method has higher success rate than using animal for fertilization. It has higher rate. It is easier and more convenient ways than to transfer real animals. This method helps to produce better quality of spring. Write the number of chromosomes found in the shell of following animals for external. This is just for external knowledge. Like mice has 40 chromosome, housefly has 24 chromosome, gorilla has 48 chromosome, eastern mucor, two different organisms, and these are one pair chromosome. This way you can learn this table. The figure shown in the type of nucleus found in the living cell. What is figure of? It is of DNA, double stranded structure DNA. What is ATG stand for? adenine, thymine, guanine, cytosine. What are the components? Ribbon-like structure, wrapped one over. It is deoxyribose sugar and phosphate molecule. Right? What is its function? Write its function. The main function of to transmit hereditary character from parents to offspring. That is DNA function. Describe the structure of RNA. RNA structure is described here. It is single-stranded. So here is everything described about it. You could learn yourself. RNA. Three types of RNA: tRNA, mRNA, and rRNA. mRNA genetic information. So all RNA are described here. Describe the structure of DNA with suitable diagram. So DNA structure is a double-stranded structures. It is described here. DNA is a part of chromosome responsible for transfer character from parents to offspring. It is helically twisted double-stranded structure. Double-stranded. So this is one strand, this is another strand, double-stranded. A DNA molecule contains two unbranched complementary strands. So everything is described here. Adenine and guanine, collectively called purines. Purines, PURINs, and the cytosine and thymine are collectively called pyrimidine. Pyrimidine. The adenine is associated with thymine by two covalent bond. There is two covalent bond. Similarly, G and C. Cytosine connected to guanine by guanine is G. By three covalent bond. This is all about the DNA structures. Everything explained about DNA structure. What are the different phases of mitosis cell division? Describe a suitable diagram. This is for extra knowledge. It's not given in the curriculum detail. So basically mitosis consists of following stage: prophase, metaphase, anaphase, and telophase. Prophase has these characters. It is first stage of mitosis, prophase. The nuclear membrane and nucleolus. Nucleolus means this nucleolus start to disappear in this phase. Chromosome becomes shorter, thicker, and distinct. Centrioles divide and move towards opposite pole. Centriole divides and move towards opposite poles. Centriole means this structure divides. Similarly, metaphase, centrioles reach opposite poles. Centrioles reach two opposite poles. This metaphase stage. Chromosomes are arranged in the equatorial plane. This chromosome. Spindle fiber are attached to centromere of the chromosome. Centromere of the chromosome. Anaphase, it is migratory phase. It is migratory phase because daughter chromosomes are formed and migrate towards opposite poles, moves towards opposite poles. Spindle fiber contracts. The centromere divides and daughter chromosome are formed. The daughter chromosome move towards opposite poles. So opposite poles. Telophase. This is the last phase. Daughter chromosome reach opposite poles. Nucleus, nuclear membrane reappear. Nucleolus this and nuclear membrane this. Round dotted structure formed. Finally, two daughter cells are formed. What are different stages of meiosis 1? Explain with diagram. This is also for extra knowledge. This is not given detail in book curriculum. The different phases of meiosis 1 are as follows. Meiosis 1. This is structure. Prophase 1, metaphase 1, anaphase 1 and telophase 1. Prophase 1 has following characteristics. Metaphase 1 has following characteristics. Chromosome arranged equatorial plane, spindle fiber arranged to centromere of the chromosomes. Anaphase 1 has following characteristics. Telophase 1 has following characteristics. Write the features of different phases of meiosis cell division with figures. This is also for extra knowledge. It's also not given detail in curriculum. So these are the different phases of meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2. This is the figure. Prophase 1 has following characteristics. Metaphase 1 has following characteristics. Aligned means arranged. Arranged in equatorial spindle fiber are produced. Metaphase 2 has following characteristics. Anaphase 2 has following characteristics. You can pause and learn slowly also. Telophase 2 has following characteristics. This is the end of this chapter. Thank you very much. The video has become quite long because it contains lots of information. It is a long chapter. Thank you for watching. If you have any question from this chapter, you may make a comment in the comment sections. So keep watching my channel. I'll come up with some other chapters, important notes, question answer as well. Thank you very much.